Hi everyone, this is Glenda Ganzon and welcome to Human Anatomy and Physiology class. And for today's lesson is still a continuation on the cellular level of organization, particularly on the transport across cell membrane, which is the active transport. So stay tuned. For all the transport method described in my previous video, which I'll be putting the link in the description box down below, the cell expends no energy. Membrane proteins that aid in the positive transport of substances do so without the use of ATP. During active transport, ATP is required to move a substance across a membrane, often with the help of protein carriers and usually against its concentration gradient. One of the most common types of active transport involves proteins that serves as pumps. So what is a pump? The word pump probably conjures up thoughts of using energy to pump up the tire of bicycle or a basketball. Similarly, energy from ATP is required for these membrane proteins to transport substances, molecules or ions across the membrane, usually against their concentration gradient from an area of low concentration to an area of high concentration. So the sodium-potassium pump, which is called as sodium ATPase, transports sodium out of the cell while moving potassium into the cell. The sodium-potassium pump is an important ion pump found in the membranes of many types of cells, and these pumps are particularly abundant in nerve cells, which are constantly pumping out sodium ions and pulling in potassium ions to maintain an electrical gradient across their cell membranes. So what is an electrical gradient? An electrical gradient is a difference in electrical charge across space. In the case of nerve cells, for example, the electrical gradient exists between the inside and outside of the cell with the use of being negatively charged or relative to the outside. So the negative electrical gradient is maintained because each sodium-potassium pump moves 3 sodium ions out of the cell and 2 potassium ions into the cell for each ATP molecule that is used. So this process is so important for nerve cell that it accounts for the majority of their ATP usage. So this is a picture of sodium-potassium pump. The sodium-potassium pump is found in many cell membranes. Powered by ATP, the pump moves sodium and potassium ions in opposite direction, and each against its concentration gradient. In a single cycle of the pump, 3 sodium ions are extruded from and 2 potassium ions are imported into the cell. Other forms of active transport do not involve membrane carriers. Endocytosis, or meaning bringing into the cell, is the process of cell ingesting material by enveloping it in a portion of its cell membrane, and then pinching off that portion of membrane, and once it is pinched off, the portion of membrane and its contents becomes an independent intracellular vesicle. So what is a vesicle? A vesicle is a membranous sac, a spherical and hollow organelle bounded by lipid bilayer membrane. Endocytosis often brings materials into the cell and that must be broken down or digested. Another is the phagocytosis, or cell eating. A phagocytosis is the endocytosis of large particles. Many immune cells engage in phagocytosis of invading pathogens like little Pac-Man, if you still know the game before, which is the Pac-Man. Their job is to patrol body tissues for unwanted matter such as invading bacterial cells, phagocytize them or eat them, and digest them. In contrast to phagocytosis, Pinocytosis, or it means cell drinking, brings fluid containing dissolved substances into a cell through membrane vesicles. So in this picture is the three forms of endocytosis. Endocytosis is a form of active transport in which a cell envelopes extracellular materials using its cell membrane. In figure A, in phagocytosis, which is relatively non-selective, the cell takes in a large particle. In the figure B, it is a pinocytosis. The cell takes in small particles in fluid. And in figure C, in contrast, receptor-mediated endocytosis is quite selective. So when external receptor bind a specific ligand, the cell responds by endocytosing the ligand. So phagocytosis and pinocytosis take in large portion of extracellular material, and they are typically not highly selective in the substances they bring in. So cells regulate the endocytosis of specific substances via receptor-mediated endocytosis. Receptor-mediated endocytosis is endocytosis by a portion of the cell membrane that contains many receptors that are specific for a certain substance. Once the surface receptors have bound sufficient amounts of the specific substance, which is the receptor's ligand, the cell will endocytose the part of the cell membrane containing the receptor ligand complexes. So iron, a requirement component of hemoglobin, is endocytosed by red blood cells. In this way, iron is bound to a protein called transferrin in the blood. So specific transferrin receptors on red blood cell surfaces bind the iron transferrin molecules, and the cell endocytoses the receptor ligand complexes. So in contrast with endocytosis, exocytosis, or what we call as taking out of the cell, is the process of a cell exporting material using vesicular transport. So this is a picture of exocytosis. Exocytosis is much like endocytosis in reverse. Material destined for export is packaged into a vesicle inside the cell. So the membrane of the vesicle fuses with the cell membrane, and the contents are released into the extracellular spaces. Many cells manufacture substances that must be secreted, like a factory manufacturing a product for export. These substances are typically packaged into membrane-bound vesicles within the cell. So when the vesicle membrane fuses with the cell membrane, the vesicle releases its contents into the interstitial fluid. So the vesicle membrane then becomes part of the cell membrane. Cells of the stomach, for example, and pancreas produce and secrete digestive enzymes through exocytosis. So endocrine cells produce and secrete hormones that are sent throughout the body, and certain immune cells produce and secrete large amount of histamine, which is a chemical important for immune responses. In this picture is a pancreatic cell enzyme products. The pancreatic acinar cells produce and secrete many enzymes that digest food, and the tiny black granules in this picture is the electron micrograph, are secretory vesicles filled with enzymes that will be exported from the cells via exocytosis. So this ends my lecture about the active transport in the cellular level organization. If you have any question, please write it in the comment section down below, and I will be glad to answer them all. And once again, I would like to thank you all for watching this video and for listening to my discussion. Until my next video, bye for now.